My name is Ana Domingos. I am the principal investigator of the Obesity Laboratory at the Gulbenkian Institute for Science, which is based in Portugal. The Institute for Science of Gulbenkian is part of a greater institution called the Gulbenkian Foundation. And the Gulbenkian Foundation is a philanthropic institution that caters to the arts and sciences. Not only we have a science institute, but we also have a big orchestra and a chorus and a museum of modern art and also a museum of not modern art. So we have the classical Rembrandts, Monets, Manets, Renoirs, and Rodins, and also a big collection of prehistoric art and also Greek art, Roman art, and a big collection of archaeological findings. There is an archaeological finding that dates to a date that precedes the invention of agriculture. And that's an archaeological finding that was found in Austria, and it's the Venus of Willendorf. And as you can see in this picture, the Venus of Willendorf is extremely obese. And some anonymous artist devoted or was inspired by this morphological variation of body shape. So as you can see, obesity is old. It has been around for a quite long time, but it's not until recently that we have a molecular understanding of its basis. The first hormone that was discovered that underlies obesity is the hormone leptin. As you can see here, we have a child that is extremely obese. This child is hyperphagic. This child has an uncontrolled eating behavior. And the reason that underlies this behavior and this obesity is the lack of the hormone leptin. As you can see here on the right, this child was cured with injections of recombinant leptin, so leptin that it's made in the lab, and this child completely normalizes. This discovery was made by Sadaf Farooqi at the University of Cambridge, and preceding the discovery of leptin in human in 1995 was the discovery of leptin in mice by Jeffrey Friedman at the Rockefeller University in 1994. So this extremely obese leptin deficient child could not be cured if one would not have found the rodent equivalent, so rodent leptin. So Jeff found this hormone in mice. Jeff was able to map the genomic locus of the hormone leptin, and ever since there has been an enormous endeavor to understand how leptin works in our body. Leptin can be called an adipostat hormone, and this resembles a little bit the word thermostat. We all know what a thermostat does. So the thermostat senses the environmental temperature, and it adjusts the temperature so that we have a very narrow range of variation of the temperature. And we can think that leptin is doing an equivalent role. Leptin is produced in the adipose tissue by amounts that are proportional to the amount of fat, and it signals in the brain how much fat exists in the body. And by doing so, when leptin enters the brain, it enters to activate a plethora of signaling pathways, which then turns into a decreased food intake, which in turn decreases the amount of fat. And with decreased amount of fat, there's also a decreased amount of leptin, which in turn increases the food intake, and which in turn increases again the amount of fat. So these type of loops that resemble a thermostat, and in this case it's an adipostat, it's a negative feedback loop. And negative feedback loops are very common in biology, and they control a variety of physiological processes, like menstruation, like thermoregulation, for instance. This is a picture of Jeffrey Friedman, whom I had the pleasure to host in a meeting in Lisbon, and with whom I had the pleasure to work for 7 years. I also hosted at that meeting Steve O'Reilly, with whom Sadaf Farooqi worked. And as you can see here, Steve O'Reilly has a lot more to say about obesity. It told us that there's a lot more human genes that have been discovered that underlie an obesity phenotype. So there's many more human genes that have been discovered in the last 20 years since the discovery of leptin. and Sadaf Faruqi and Inês Barroso have been driving this discovery. And a lot of these genes that we can see here on this list, like SIM1 or MRAP2, which have been published in journals like Science or Nature or Cell, a lot of these genes, we have now a glimpse of what they're doing, and perhaps they could be a good therapeutical entry point to address obesity. There's a list of genes for which we have some physiological insight, but there's a growing list of genes that are now being mapped out. So here we see in this data from Sadaf Faruqi, Steve O'Reilly, and all, we see that there's a huge list of genes, and if not genes, those are point mutations in the genome that are indicative of an obesity phenotype. So if a patient is obese, a patient in principle would have the right to know whether or not they're mutant of these genes, or whether or not their genome has as the mutational signatures that are indicative of obesity, particularly if we're talking about people who are obese and they have been obese ever since they were born or since they were very young. So those mutations, if they cannot be indicative of a therapy, at least they can be indicative of a diagnosis, whether or not these people have morbid obese that cannot be cured by behavioral therapies. So this is the genetics of human obesity that has been pushed forward by the group of Steve O'Reilly at University of Cambridge. And there's a lot of endeavor that has been made trying to understand how do you go from a point mutation to a whole body obesity. And that's related to how do cells work. A technique that has been a major asset in understanding leptin action in the brain is optogenetics. As you can see from this word, the prefix of the word is opto, and opto means light. And light travels to optical fibers. You all may know about this because everybody has like high-speed internet at home, and we all know that it goes on an optical cable. Optical cables transmit light, and I'll get into the details of how can this help us understanding the biology of obesity. Optogenetics has been popularized in the last 10 years by Carl Deiseroth at Stanford University. And we see here that there's a number of publications that have been popularizing this technique, and it's a growing field of research and utilization in applications. As you can see in this picture, we have a rat that has an optical fiber that it's penetrating deep down inside its brain. And I have to make a statement that none of these technologies or these procedures are painful to animals, and there are ethical committees that are involved in making sure that none of this causes distress or pain to mice. And one of the reasons why it's painless to have an optical fiber implanted in the brain is that the brain does not have pain receptors. Moving forward, what is optogenetics? We know about the light, so what about the genetics, which composes the word optogenetics? Optogenetics relies on a gene named chanorodopsin, which was isolated from deep sea algae, and that's seen here on this picture on the right. These are unicellular flagellated animals that live deep down in the sea. And deep down in the sea, the only wavelengths to penetrate are wavelengths that are similar to blue. So blue light penetrates deep down in the ocean, and these creatures, they move the flagella towards the light so that they can swim towards the light. This is a process called phototaxis. And they do that because they have a gene in their genome that is a cation channel. This channel sits in the membrane, and when this channel sees blue light, it opens and it permeates cations. So those are ions like potassium and sodium. And when it does that, the flagella starts to move. Well, if we placed this cation channel, light-activated cation channel, so chanorodopsin, in a neuron, this is what happens. We can activate the neurons with flashes of light. In this picture, we're looking at a mouse brain that has received chanorodopsin transgenically, so we can use transgenic techniques to deliver chanorodopsin into neurons. And you can see that here in this green spaghetti that we see here in this picture. There's a subpopulation, a very specific population that we can deliver this channelrhodopsin. And when we provide light flashes of blue light, we can turn on the neurons at will. Here we see an example that you can turn on the neuron at 5 hertz. You can also turn on the neuron at 20 hertz. We can turn on the neurons at whatever frequency one desires to. We can verify that turning on the light deep inside the brain actually activates neurons. In this picture, you see a brain, a rodent brain, and you can actually see the fiber tract of the implantation. We implant deep down in the brain the optical fiber. We can cement it so that it's secure and it doesn't move, and we can turn on the light, and you can see that the neurons only get activated when the animals have channelrhodopsin, but the neurons do not get activated if the mice do not have the channelrhodopsin. And we can turn on and off the neurons at will. And why is it good for? Why do we do this? We do this because we have questions that we want to answer. And the questions that we want to answer are, what are the neurons in the brain that are controlling appetite and that are the targets of leptin action? Here we can see a movie that answers that question. In this movie, we have a mouse, and this mouse has channelrhodopsin in a very specialized subset of neurons called the AGRP neurons in the hypothalamus. So that's really deep down in the brain. And when you turn on the light in these animals, you can see the light flashing across the fiber. The animal is sleeping, but as soon as we turn on the light, we see that the animal suddenly wakes up and starts binging. There's pellets here on the left side of the image that are available to the animal. So pellets are mouse chow, and the animal wakes up and it starts eating. And he does this over and over and over again until the light stops flashing. So this demonstrates that appetite is not really under voluntary control, but it is under control of neurons that are sitting deep down in the hypothalamus and that are very far away from voluntary control. So what we can think is that if we could pharmacologically activate or deactivate these neurons, then one would be able to control appetite. That's in the brain, but the brain is very difficult to target for pharmacological reasons. There's very few drugs that are able to cross the blood-brain barrier, so this is a physiological barrier that it's well known in the pharmacological development that prevents from drugs entering the brain. So another solution or another idea or avenue of research would be to avoid the brain to target obesity. And this has been a focus of my lab. In this slide, we see this is a textbook. It's written in German. I don't read German, but nevertheless, the picture tells you what I'm focused on. This is an neuroanatomical drawing of the sympathetic nervous system. And as you can see here, there's no brain in this picture. And what you see here is that there's a variety of neurons and a huge population of neurons that has been largely understudied. And this anatomical book that medical students read, you see that there's a couple of problems with these images that have been copied generation after generation since the mid-1800s. So there's really not that much molecular detail into these images, into these textbook images. There's two features that are a bit worrisome about this picture. The one feature is that all of these neurons, they're labeled with the same color as if they were all the same. And that's maybe or perhaps not the case. So that's one point. The other point is that there's no fat to be seen in this picture. And there's no fat to be seen in this picture, although fat is an endocrine organ that secretes hormones like leptin. Everybody knows how to draw a heart, a pancreas, or a liver, but nobody really knows how to draw the fat organ as an organ. And our lab tried to address that question. So in this picture, you see a piece of fat that has been subject to two techniques. One of them is called OPT, optical projection tomography, coupled to tissue clearing. So basically, we render transparent a piece of fat, and then we image this piece of fat. And this is the image that we get. As you can see here, this is a piece of fat that has a lot of complexity. It has a vascularization, it even has a lymph node, and there's elements in this picture that reveal to us that, in fact, the adipose organ is innervated. We can segment and we can take a closer look at these nerves that you can see here in this picture, and you can see that there's axon bundles entering the adipose tissue. We can in fact dissect these axon bundles, and we can subject them to various procedures to try to understand what axons are innervating, what kind of axons are innervating this fat depot. And as you can see here in this picture, we have red, let's say, spaghetti. So the spaghetti is basically axons that have a certain molecular identity, and the molecular identity is sympathetic. So we know that these neurons are sympathetic neurons, as opposed to any other neurons that are labeled green that we don't really know what they are. And this is important to know what things are. We can use transgenic techniques to follow these neurons and ask where do they end up. And we can see that the sympathetic neurons here in this picture that are red, they're able to target adipocytes. So for the first time, it was demonstrated in 2015 by my lab that adipocytes can actually receive neuronal innervations, sympathetic innervations. And the question is, what are they doing there? And to answer this question, we have to do what in biology we call a gain-of-function experiment. This is just fancy wording for saying that we want to activate these neurons to see what's the outcome. And again, we take advantage of channel-rhodopsin to activate these neurons and ask what happens to the adipocytes when we activate these neurons. We can implant an optical fiber that targets the neurons deep down in the fat, and we turn on the light and ask what happens. What happens to the fat that has been light stimulated versus the fat that has not been light stimulated? To answer this question, we stimulated the mice only on one side. In this picture, you see that there's a patch of fat that has been labeled blue versus a contralateral control side that has not been flashed and therefore is labeled yellow. And we place these animals inside an MRI machine so that we can measure the amount of fat in one side versus the other. And what we saw was that animals that do not have channel-rhodopsin but were subject to the same experimental procedure, so they were stimulated on the right side but not on the left side, the fat pads on either side, they look pretty much the same. However, if animals have channel-rhodopsin, we see that animals that were stimulated on the right side, they have a marked reduction in the amount of fat versus the amount of fat that it's placed on the contralateral side, so on the left side. So this tells us that local optogenetic activation of sympathetic neurons reduces fat locally. And the word local here is very important because we want to have a therapy that targets only a subset of cells that it's very small, but a subset of cells that does the right thing. And the right thing is to promote lipolysis. It's to promote fat reduction. Now, can we take advantage of this biological mechanism to develop therapies? The answer is yes, and this is something that my lab is actively engaged. We know that amphetamine is a very old compound that has a very potent anti-obesity effect. And everybody thinks that amphetamine acts in the brain to suppress appetite and to promote locomotion. So that's called hyperkinesia. It could well be the case that this is not the full story. And amphetamines, they've been coined as sympathomimetic. So that's a very fancy word for saying that activates the sympathetic nervous system. And people think that this is the case because patients who are under amphetamines, not only they become addicted, which is a very big problem, but they also have increases in arterial blood pressure. So this is considered a side effect, but maybe these side effects, they can be circumvented so that we have a specific action in the sympathetic neurons that innervate the fat. We're chemically modifying amphetamine so it doesn't reach the brain, so that amphetamine is not anymore an addictive substance. And we're also modifying amphetamine so that it can be targeted to the sympathetic neurons that we care about, and those are the sympathetic neurons that innervate the fat, but not sympathetic neurons that may control arterial blood pressure. So this is a glimpse of what I think it's the future of obesity research, but many other people think that the future goes in various directions. And there's a number of journals that are dedicated to understanding and pushing forward the therapeutics of obesity by tackling down the biological basis of obesity. And this is just an example on this slide where we can see various journals. We have molecular metabolism, cell metabolism, journals of the cell press group, Nature. As you can see here, all of these journals care very much about understanding the biological basis of obesity, and that's really the only hope for patients. And as Stevie Wonder said, when you believe in things you don't understand, you call that superstition. And science is not about superstition. Science is about understanding what pharmacological compounds do. And for that, we need first to understand what is the biology of obesity so that we can identify cells that we can target, and we can identify molecules in those cells that can be targeted with a drug. Science is not about superstition, but there's a lot of superstition out there that it's still deceiving patients, and unfortunately in a bad way. If you type in a Google search for weight loss pills, there is a variety of companies that are selling products that really do not work. As you can see here, there's a variety of products that have been advertised by male white doctor, and it targets people that really are not obese. It targets people that they think that they can have a body like Barbie, whereas, as you can see in this picture, if Barbie had the natural morphometric features of a regular average woman, Barbie would look very different. And this type of perverse body shapes has been a constant throughout history, and they're nothing but torture. In this case, it's psychological torture, because you convince women or men that they can have a body shape that it's totally not natural, and that has been observed across the ages. So here on the top right corner, you see a lady that had a wasp waist, and of course, wasp-like waists are not natural, and that has caused a lot of suffering for women in the mid 1800s. There's abnormal head shapes that have still utilized this day by some ethnical groups, and of course the Asian super small feet that in reality caused a lot of physical pain and also psychological and social control. So a body shape like Barbie is no different from the various forms of torture that have been socially imposed onto women. I have to thank my group, my lab, the Obesity Lab at the Gulbenkian Institute, and I also have to thank my funding agencies, including the MIT, the Massachusetts Institute of Technology, who is that my student Roxana belongs to. She is a PhD student from the MIT Portugal program. The European Molecular Biology Association, the FCT, which is a Portuguese funding agency, of course, the Carlos Gulbenkian Foundation, and just recently we were awarded with the Human Frontiers grant.